There'll be a short presentation, like really short. The main goal of this is to ask questions and get some qualified answers. Andrew Jackson is our commercial director. He's based out of Budapest. He's currently in Boston. He'll be in the United States this week through the middle of next week. And I'll allow him to introduce himself in just a minute. My name is Kevin Bittman, and I am a content manager with Syngenix. I came on board on August 4th is when I started. And prior to that, I worked for Agilent as part of their cell analysis division. I was with Seahorse, and I did a lot of work with metabolism. I actually started with Seahorse in 2008 as a field applications scientist. I really enjoy telling stories, and I got to do a lot of presentations. And in my role as a content manager, I get to tell some stories, get to write some stories, and that's what led to the poll. There's a button at the top of the screen that says polls. If you click on that, you'll see a question. And the question is quite simply, when was the word antibody first introduced into the literature, or when was it coined? I'd love to hear from you. And all the dates that are listed there are pretty important dates, actually, in terms of antibody development. I've been going through a history of antibodies, trying to learn a lot of stuff, and I'll be writing a white paper on that. And I think it's going to be really interesting and pretty fascinating. So I came on board Syngenix in August, and Syngenix was born out of some research that was being done by Professor Blackburn, who was at Cambridge University back in 2008. He's now in South Africa in protein chemistry, and basically designed techniques for capturing fully folded functional proteins onto streptavidin surfaces. So we can print these onto a microarray, and we have a fully folded, properly conformed protein, which is going to improve your antibody binding. And we know that indirect immunofluorescence is one of the main ways of studying protein expression and for doing protein microarrays. Another common way of looking at proteins is mass spec. The technology was put into factory production in 2016, and we are supported by Summa Equity, and we'll be opening, actually, technically it's open, but I don't know if anybody's in there yet, our office in Boston. We have the space, it's been renovated and ready to move in 2022. So I also want to point out, and Andy had mentioned this while we were talking before, we have a really exciting poster at the Society for Neuroscience. So if you happen to be going, November 13th from 8 a.m. to 12 p.m., it will be Man by Dr. Garnett, and it's poster F23, 121.2, Parkinson's disease. We coupled with the Michael J. Fox Foundation, and we've identified novel autoantibody signatures. So these are biomarkers that can help you identify Parkinson's and the likelihood that Parkinson's will progress into deteriorating symptoms. We also will be present at booth number 621. So if you can't make it to the poster and you want to talk to some Syngenix folks, head over to booth 621. We'll be there all week. Andy will be there as well. Now, this is a webinar that we've put together. This is the first one that I've run, and I will be running more of these in the future. Likely we'll do one every other month, and if you've got any suggestions of things you'd like to learn about in terms of protein chemistry, microarray, biomarker discovery, and so forth, let us know. What we have planned for the future is autoantibodies as biomarkers, why it's such an important field, biomarker discovery, and how autoantibodies can really play a profound role in the development of these new biomarkers for diagnostics of diseases such as cancer and neurodegenerative diseases. We'll be talking about autoantibodies in cancer. Not sure if you're aware of this, but all the way back in about 1910, folks started to notice that there were antibodies in the sera of patients that had malignant tumors. So it's been around for a while, and we're taking advantage of it now because we've got the technology to do so. And part of that technology involves bioinformatics, which is another topic we want to talk about. This is the ability to take large chunks of data and then find correlations across those large chunks of data. It's interesting because bioinformatics isn't just about science anymore. In fact, it started out kind of as artificial intelligence, but you'll see it if you watch Sunday football now. They use bioinformatics in order to determine what's the likelihood of that guy's going to catch a touchdown. I don't watch European football so much, so I don't know. I'm sure bioinformatics is in there too, but I don't know if they have all those crazy diagrams like they do with the American NFL. And we'll also be talking about neurodegenerative diseases and autoantibodies in a future session. So with that, I'm going to jump right on in. And what I've done is, as I've been learning about the technology and autoantibodies and the immune system in general, I've come up with a number of questions. I've even reached out to some people, maybe some of them are on the call today, asking questions about what role antibodies and autoantibodies have played in disease over the years and how they've been used as biomarkers and so forth. And I've come up with this number of questions, and Andrew was kind enough to help me with answering these questions. So if you have a question, like I said, put it into the chat window and I will ask it of Andy. But for now, I have, let me stop sharing my screen. I have questions that I put together for him, and there we go.
If I can perhaps just do a quick intro as well, Kev, I'll speed up. So good morning, everybody. Good afternoon. Andrew Jackson, Commercial Director, US. My background was initiated within the big pharma world, so working for Novartis, J&J in various commercial capacities. I actually supported Sengenics in terms of the commercialization of the technology around in 2017 now, and have been part of the journey through our successes with big pharma, through the academic world, etc. So hopefully we can share some of that experience with you today as we sort of answer the questions.
Thanks, Andy. All right, so that was a little bit of your background. One of the first questions I had is, how did you come to Sengenics?
Relatively fortuitously. I was working in the microarray field at the time, and I met Jonathan Blackburn, our CSO, fortuitously down in Cape Town in South Africa, and conversation developed from there. Jonathan started to speak to me a bit more about the concept of autoantibodies, and I think that's also as part of today's discussion as well, really important to define what is an autoantibody. I think there's a lot of misconceptions out there around what autoantibodies do, the role that they play in particularly disease manifestation, and also the types of diseases that autoantibodies are present in. So I know many of you know what an autoantibody is, but just kind of for definition's sake, an autoantibody is an antibody that's made against a substance, which is basically formed inside one's body. And the autoantibody has the ability to either directly destroy a cell, or it can make it easier for your white blood cells to destroy that specific target in the body as well. And as I mentioned, there is a misconception that autoantibodies are only prevalent inside some of your classic autoimmune diseases, like your lupuses, your RAs, your Sjogren's, your IBDs of the world. But autoantibodies actually play a prevalent role in many other different diseases, particularly in cancers, particularly in neurological disorders as well, such as Parkinson's, Alzheimer's, and the alike. So just want to make that really clear, because it sounds to me, speaking to the field, at times people get a little bit confused, and it seems to be a relatively closed term, and a term which is very much dumbed down as being pigeonholed into certain types of diseases. And that's not true. Autoantibodies are prevalent across a whole multitude of different diseases, indications, and the alike, really. So just wanted to get that off the table first and foremost.
Yeah, and I think a lot more research needs to be done. There are some circumstances where autoantibodies may be beneficial. There is a bizarre paper that came out not too long ago on the bubonic plague, and they had basically demonstrated that people that had lupus were more likely to survive the bubonic plague, and that wound up in the genetic history in the genetic tree. So there's actually a gene associated with it. That they don't know why. They don't know if maybe this autoantibody may have been a byproduct of trying to fight bubonic plague that wound up being beneficial for these people. If there's something more to it, obviously they used bones, the remains of people, to do these studies, so it's difficult to tell. But really some interesting stuff, and I think more research needs to be done about autoantibodies beyond just these autoimmune diseases, as Andy had mentioned. So could you tell us a little bit about seromics and why it's important and who is doing this type of work?
Yeah, absolutely. So, obviously seromics is the totality of antibodies within inside a serum sample. And I will say, just from our perspective, the majority of the work that Sengenics are doing are with serological plasma samples, which are highly enriched with autoantibodies, which gives us a lot of optionality in terms of being able to A, measure them, B, detect them, and C, start to manipulate and further understand the functional role of those autoantibodies within specific patients. But fundamentally, being able to interrogate a serum sample gives us a real live snapshot of what is happening inside the human immune system. So the humoral human immune system is obviously constantly surveilling and making aberrant protein changes during disease manifestation, during progression, whether that be during a period of treatment or non-treated. There is a constant evolution of the autoantibody and the state within the body that we're able to measure, which is a really, really good opportunity for us to, again, understand, the likes of the lupuses of the world, where patients experience flare-ups, which are very hard to track and understand, humorally what is happening during specific types of events within disease. But I think also just to be clear, autoantibodies are biomarkers which are also orthogonal. And I think there's always, again, just to sort of highlight a misconception there. People tend to think it's an either or when you start to look at biomarkers, but I think the key message really to everybody is that biomarkers should be complementary. And certainly we have a number of papers coming out now, and certainly some of the strongest findings in recent years around cancers in particular, and also autoimmune, have been very highly enriched multi-omic sets, and that means that autoantibodies have a potential role to play in that as much as possible. But it certainly gives us biologically relevant insight into disease state, and I think that sort of leads us into our sort of next question, Kev.
Right. I got to get used to the sharing of the screen. Okay. So the next question really was about Syngenix, and where does Syngenix fall into all of this?
Yeah, absolutely. So, I think there has been a dogma in the field, which has not necessarily been addressed historically, and that dogma has been that autoantibodies are extremely powerful biomarkers. By definition, and by nature, they're highly specific. They're abundant within the human blood. They're also highly accessible and also very stable. Which means that gives us a lot of opportunity to go back and interrogate samples for different types of analytical approaches. But also in terms of the way that autoantibodies interact. And one thing we do know from the literature is that autoantibodies bind to conformational linear epitopes. And just to sort of put that into perspective and fundamentally how this is linked to the Syngenix technology is that for autoantibodies to be able to bind effectively to the target of specific interest, all of the epitopes on the protein of interest have to be preserved and presented in a biological and functional manner. And this means that the protein has to be correctly folded. This again will give full exposure to all potential epitope targets. And if the protein is not fully folded, and this is kind of where the challenges in the field have arisen over the last number of years, where previous technologies, some existing technologies out there, have been deploying partially fragmented proteins, partially folded, which fundamentally exposes non-hydrophobic surfaces, which many of you out there who have used protein technologies in the past have probably experienced that problem, which consequentially ends with non-specific binding, and we end up getting biologically irrelevant data. The Syngenix technology has been able to address this very eloquently by ensuring that we have a methodology using a fusion protein called biotin carboxyl carrier protein, BTCP for short, which basically acts as a solubility enhancer. So when our protein of interest, we've cloned and expressed thousands of human proteins now in our library, from signaling molecules to kinases to different types of cancer-testis antigens. When the protein of interest is correctly folded, sorry, I should have said the protein of interest is cloned in frame with this BCCP. And when the protein is correctly folded, this co-translationally drives the correct folding of the light blue BCCP, the biotin carboxyl carrier protein, and gives it a mechanism of being able to bind to the surface through autobiotinylation. And the surfaces of our arrays are bound with streptavidin, which has a very strong binding affinity with biotin itself. And in contrast to that, we also find that when proteins are misfolded, partially folded, this drives the misfolding of the BCCP and is unable to autobiotinylate itself. So fundamentally, to summarize that, we have an extremely powerful method of ensuring that only correctly folded proteins are bound to the surfaces of the arrays. And this has allowed us to really negate the issues of partially folded proteins. Again, as I mentioned, the way that autoantibodies behave is binding through conformational linear epitopes. So we've been able to provide the scientific community with a real step change in technology, ensuring that we get biologically relevant hits. And we're not going to talk about bioinformatics today, but certainly through this methodology, conceptually, that's how we've been able to address a real big need.
Thanks, Andy. Okay, so you did touch on this a little bit, but I was hoping you could go into some more detail. How do we take advantage of autoantibodies in our research or across different research disciplines?
Sure, that's a great question. I think, you know, really sort of trying to address the sort of current needs within the scientific community right now. Firstly, that being trying to understand disease state and trying to understand heterogeneity with again across a whole multitude of different diseases, whether it be cancer, whether it be autoimmune, or whether it be the likes of COPD, for example, which is extremely heterogeneous by nature. We have developed a number of applications using the protein-based array technology, as I just described, which allows us to firstly go through discovery-based projects, whether it be through profiling to identify potentially autoantibodies on a highly enriched panel of antigens, to discover potentially autoantibodies that are driving pathogenicity, trying to understand and determine the functionality of the role with those antibodies within a disease. But from that as well, trying to find markers that potentially have diagnostic value, prognostic value, or in some instances, and I know you alluded to this earlier, Kev, with your analogy around the plague, even having protective autoantibodies as well. And we're certainly involved in research right now, particularly in the neurological field as well, where we are, you know, really trying to potentially identify protective markers for some of these types of diseases as well. And there we go. We've got a couple of different case studies there. But I think sort of just moving that along, that would be the first application where we're really able to address a burning need from the community. I think secondly, and this is where we sort of step into more of the clinical realm, using designs. We've designed numerous projects now as well, where we've been able to set up experiments to look at intra-disease stratification. So really trying to understand subpopulations of specific diseases, again, across a whole multitude of cancers, neurological, autoimmune, and the alike, infectious disease, and really using that for kind of reverse translational approaches. So using some of the existing treatments and therapies that we have to try and identify subpopulations within those cohorts. And sort of moving that further along, probably the third application would be around response prediction. So being able to predict by utilizing and discovering signatures correlating to response, or in some instances non-response, to support in terms of developing assays for enriching decision making, for patient selection, which is certainly becomes even more of a challenge as we start to gravitate through to your phase 2, phase 3 trials, and And also alongside that as well, our ability now to predict immune-related adverse events in the cancer field, which for those that are working for big pharma in particular is one of your major headaches right now. And from that, not only predicting the IRE, but also predicting the actual organ-specific IRE. So again, allowing individuals who are going through clinical trials to give more visibility to clinicians around the potential to develop an IRE, and really allow clinicians to monitor those patients whilst they are on a treatment, and if necessary, prevent them from developing a high-grade event, which again could cause a multitude of different issues to the patient, potentially death. So that's kind of the crux. And I would say there's also a fourth as well, which we don't speak about too often, but we've certainly been heavily involved in from a vaccine perspective, where not necessarily autoantibodies, but we have been using the platform to develop custom arrays specific to vaccine programs, where we've been able to develop specific targets, not just kind of your simpler, well, I wouldn't say simpler, it's not easy to produce any kind of protein, but perhaps producing more complex types of structures like heterodimers, tetradimers, and really giving vaccine developers an opportunity to screen for spectrum of neutralization. So again, that's slightly off topic to autoantibodies, but certainly another way in which the technology has been utilized. And I will just stick on the vaccine topic, which is specific to autoantibodies. We also have been very active in terms of the COVID developments in recent years as well, where we've actually developed a COVID array, which has over 50 different targets on now, including a whole handful of different Omicron variants, and particularly looking at understanding the potential for new therapeutics to prevent hospitalization. Obviously, I know COVID is a non-defined term, really, from a clinical perspective at this moment in time, and really supporting, our collaborators and our partners to understand maybe long-term ramifications of COVID at more of a biological level.
So there were a couple of things that I came across recently. I was fortunate enough to attend a biomarker consortium conference that was sponsored by the NIH last week, the week before. Yeah. And one of the things that came out of that is that the FDA and the EMA are more likely, or at least there was a higher percentage of drugs that were approved that had been through some sort of biomarker screening before they got to the approval process.
Sure.
So biomarkers play a big role in drug development, and that wasn't there, say, 10, 15 years ago. In fact, the pharma industry was suffering between 1990 and around 2010, trying to develop new drugs and new ways of making things more efficient and more efficacious and safer. Biomarkers played a role in helping to turn that trend. Dimitri did ask a question and Katie answered it. Katie McGurr is actually my boss, the director of marketing. And the question was, how do autoantibodies see intracellular proteins? And in case you didn't see the Q&A, the answer is that intracellular proteins are exposed when cells die, and they're part of the disease process. So they can get out of the cell, be recognized, and then they can be tagged by autoantibodies. Okay.
Exactly. And if I could add an extra point onto that, Kevin, you mentioned sort of the evolution of biomarkers and how they've sort of developed. And I think one of the things which I see with regularity is the fact that getting sort of new therapies and drugs onto the market isn't necessarily as much of a challenge. It's it is more about the patient selection piece. Is how can we build tools and models to support clinicians in terms of being able to identify the patient population that treatment could be more geared and effective against.
So hopefully we've made a good point that biomarker discovery and identification is important for disease, for drug development, and so forth. What about autoantibodies? Is investing in autoantibody research a risk? If you're already used to doing, like everybody's been doing genetics for like, well, I say from the 70s, but well before that. But the technology is really advanced through the 70s and 90s and the 2000s. So what are the advantages of using autoantibodies? What are the benefits, and is it risky?
So I think there's a whole raft of different responses to that, and I think overarching answer is it's not risky at all. No, I think autoantibodies have been extremely well versed and described in the literature. I don't think this is anything new. There are thousands of papers out there which explain the role that autoantibodies play within disease. I would also say as well, from a financial perspective, I think the bar is relatively low. Obviously, I know the cost of genomics has decreased over the course of time, and we're also seeing that in proteomics as well. So I think the level of entry cost is also relatively low, and I think if when we enter into discovery projects with collaborators, we don't require thousands of samples. We can work with sample ranges from 1 to 200 and come out with extremely convincing and powerful, robust data sets, which we can then look to validate and take that step further. So the ask is small in terms of patient numbers. The sample required, given the high sensitivity of the Sengenics platform, for example, is very low. I mentioned previously, we're working predominantly with plasma and serum. We are working with the likes of CSF, lung lavage, saliva, urine. We've worked with a plethora of different biological samples, but given the access to plasma and serum, we're running typically at around 10 microliters to run one of our assays. So the sample ask is small. The entry cost requirements, I think, are very modest, and I think they're extremely well versed in terms of how they've been described historically, and I think that's also synonymous with what we've seen in the scientific field. So we go out and we speak to people about autoantibodies, and there is a real trajectory now of people starting to change their mind about how autoantibodies are potentially perceived as being like a luxury or a novel type of biomarker to becoming more mainstream and really starting to understand how they complement some of the other types of technologies and approaches, the likes of genomics, metabolics, and the alike. So I think there's a real transition now, and I would certainly urge people to really think about that. I don't think there are any fear factors around that now. And as I say, probably the most prominent point is that we have a technology now, which is maybe where people have been let down and had their fingers burnt historically. They've used technology which has not been designed for a specific purpose. Fragmented proteins tend to be used for antibody specificity. This is not what the Sengenics platform is built for. It's built for autoantibody detection, measurement in a quantitative manner. So the fact we have a technology now, again, gives a real boost to people's belief and confidence.
And that sort of leads into my next question, which you kind of sort of answered, is why now? Aren't protein microarrays noisy compared to looking at DNA and RNA technology?
Yeah, and I think it's a good question, Kevin. I think people have seen that previously. I think people have looked at other types of technologies which again have employed fragments of proteins, partially folded proteins, etc. And you're going to see non-specific binding, and you're going to see hundreds and hundreds of hits within a patient, which is biologically meaningless. And I think what people are observed and people are always really pleasantly surprised about is that when they do see the Sengenics data, is the fact that we're only getting a seldom number of hits, because we're getting biologically relevant hits. We're getting 15, 20 hits maximum when we're looking at specific, autoimmune diseases, lupus and the alike. And the reason is because that's physically what is happening within the patient. We shouldn't be seeing hundreds and hundreds of hits. That's not representative of what is happening within that patient. The patient would probably be dead if I'm honest with you. So I think we've seen a real step change, and I think people are pleasantly surprised. And the fact that we're able to also correlate the findings that we often see, the likes of the lupus with your SSB and TROV2, we're seeing common reproducible hits as well. Again, I think that gives people confidence. But certainly the fact that we have, I mentioned correctly for the proteins, but also the protein chemistry, which gives very low signal to noise ratio, means that we're getting very easily interpretable data as a consequence.
So that leads into the next question, which,
Sure.
and this takes advantage of your field experience. What kinds of objections do you hear?
Yeah, I mean, first and foremost, I do hear people that say that they've used autoantibody based technologies before, and they've had maybe invariable experiences. And when I've really started to sort of push on that point and start to understand the type of technology they were using, etc., how many samples were you using? And I often hear I used 5 samples, and I'm making a decision on 5 samples. That's not good science at the end of the day. So I think really just educating people on A, the technology evolution, B, in terms of being able to, drive experiments that are well powered, and will give valuable data back in return and answer specific scientific questions, I think is one thing. And I think obviously there is always a misconception around cost, and data interpretation and all the things that sort of go hand in hand. But as I mentioned, the sort of the cost of entering into this type of technology has dropped dramatically now. And from a data perspective, I understand that autoantibody data interpretation is very much different to the likes of genomics, but that's why we're here. And we have a robust bioinformatics pipeline and team at Sengenics to support all of our customers and all of our projects in moving through that process.
So, sort of in keeping with the previous question, what do you think drives acceptance of autoantibodies as biomarkers?
What drives acceptance? I think trust. I guess every time there's an opportunity to maybe bring a new technology or a different modality of biomarker, any scientist out there will certainly ask, what is the reason to believe? Why should I use that? What value is it going to give me that I am not already being served with through some of my existing capability? So I think firstly is, we all have a responsibility, I think, as a scientific community to continue to share as much information as we possibly can through publication, through going to events and presenting data that's exciting, that can add value to the overarching goal, which is being able to further understand disease, develop therapeutics which can best serve the patients that ultimately we're trying to treat at the end of the day. So I think continually developing our publications, keeping them updated, making them relevant, expanding the range of diseases that we're very much focused on. It can be very easy to get caught up in your major cancers, given the size and the impact they have, and some of your key autoimmune diseases. But there are a lot of other indications out there which have an autoantibody component, and again can really give other individuals an opportunity to further learn more and make an impact as well. So I think we all have an obligation to publish and to keep driving the level of intelligence of the field forward as much as possible.
And I wanted to point out that NCI sponsors a group called the Early Detection Research Network, EDRN. So I had to put it in my head. And these are labs mainly within the US. I think there's one in Canada, there might be one in Europe, that basically are searching for biomarkers. And a number of them are looking for autoantibodies as potential biomarkers for cancer. It'd be huge if you could detect cancer long before it shows any symptoms. And that gets me into another question about studying autoantibodies. What techniques are available? What do you think is the best approach?
Well, certainly, the main approach right now is obviously a protein-based array approach, which correlates to the technology that Sengenics provide as well. There have been obviously other opportunities to perhaps morph into kind of bead-based assays as well. I think this is very much at its infancy right now from an autoantibody perspective. porting our technology onto different types of other types of platforms, particularly given the impressive content library that we currently have as well, provides other opportunities as well. But I think right now, focusing, given the exquisite levels of sensitivity, specificity, the reproducibility we see from our platform as well, I think protein array is the way to go as we stand today.
So again, taking advantage of your experience in the field, where do you see autoantibodies most often used in research?
Do you mean from a disease perspective?
Disease or application, yes.
Yeah. I think it really varies on who we're talking to. Right now, if we think about big pharma are all working on your lupus, your RAs, your Sjogren's, your IBDs, the likes of scleroderma as well. And particularly from the cancer perspective, we're seeing a lot of traction, particularly within prostate, melanoma, colorectal cancer. And I think that's a really interesting one if you start to think about the relationship with colorectal and IBD. There seems to be some real focus there. And also, the likes of breast and lung as well are extremely prominent. But if you start thinking about that from an application perspective, what we're seeing evolving massively now within pharma is particularly the IRE response prediction. There's an alarming percentage of individuals who die simply from the drug, some of these cancer immunotherapies, and I think there's a real focus now with such a high percentage of individuals eliciting some kind of IRE to really build tools to really support clinicians in being able to make these decisions as much as possible. So I think really that's where we're seeing a huge focus now. And then, perhaps more from the academic perspective, we're seeing a lot of traction around really trying to identify markers that have prognostic, that have diagnostic value from that perspective. So really much fundamentally more focused on disease for obvious reasons.
So you touched upon this a couple of times. I'm going to ask it directly. How does this autoantibody information help reduce apprehension and decision making, especially in the clinic?
Well, as I mentioned to you, as we sort of defined before, what an autoantibody is, how it behaves, how it manifests, how it, and what fundamentally what value that can bring to the table from being able to understand disease state. But one of the things Sengenics are geared towards, and I think this really complements where the market is focused right now, is not just using large panels to screen for thousands and thousands of targets, but really trying to identify signatures and trying to identify specific markers which can then have an impact and fundamentally some clinical utility. So again, our ability to be able to customize assays, deploying signatures and specific targets which are of relevance to a particular program or a particular scientific question is what people are fundamentally asking for right now. So we have the capability to go through discovery, through validation, through to a specific assay which can then be deployed into a program. And again, it's all on the same platform.
Nice. Now, Xavier had another question. Katie attempted to answer. I don't know the answer to this one either, but I'm not sure if you do any, but I'll put it out there.
Sure.
Xavier wanted to know if we had data comparing levels in the same sample using serum and plasma. I don't know. Katie said she was going to look into it. I don't know if you have anything to say about that, if you've come across that in your field work.
Yeah, so we do have some data to compare. I can't really say too much in terms of specifics, but they're very much similar. If you were to give me the choice between running a plasma or serum sample, my response would be either. I think we can extract the same quality, the same data fundamentally from both. I don't think we have anything that we would favor one or the other. Yeah, they do, Xavier. Yeah, they do correlate. Absolutely.
You also had mentioned this earlier, and I just wanted to bring it up again in case you had more to say about it, and that is the economy. How economical is autoantibody screening compared to other techniques, again, such as genomics?
Yeah, so, so I think you've got the financial, economical side of things, which is, um, you know, there I think, and maybe this is where the slight bias comes in. I think the thought process is that genomics maybe is a little bit cheaper at this moment in time. Proteomics is slightly newer in terms of some of the newer technologies that we have in the market. But I think in terms of what you can do with proteomics, certainly the ability to do multiplexing, high throughput, it gives you a lot more optionality with what you can interrogate with your samples. And that is why we've seen, I think, a big shift change towards proteomic technologies over the last number of years.
So getting back to Syngenics and what we do, what do customers tell you about our technology?
I think certainly I think fundamental to the things that we're doing right now is being able to have the right content. So, you can have the best samples in the world, you can be asking the most intelligent questions in the world, you can have the best performance of technology and the like. But I think comes down to content and having content specific, which will allow researchers to really identify the signatures, really to use their samples to highlight the targets which are maybe playing a prominent role within particular disease. So I think we've done a fantastic job with Syngenics at developing a library now of nearly 3,000 targets, and that's also something that we can share with people if people want to see the protein library that we currently have. We're able to do so. So I think from that perspective, content is king. I think in terms of the performance of the technology as well, we have a highly reproducible platform. We're getting piercing correlations of 0.9798. We're getting coefficient of variance of under 5. So I think that from that perspective, but I think also equally as importantly as well, the level of sensitivity and specificity. We're running at 4 to 5 orders of magnitude, fully quantitatively. And as I mentioned, in terms of the concept of having correctly folded proteins, which is so imperative to being able to identify, specific or expose epitopes which are specific to particular antibodies, is key. So that's why we see that the high specificity. So performance, I think people are enthralled by the level of performance that we have on our platform. And I think when you start to think about the signatures, the MPVs, PPVs, for the likes of response prediction that we're discovering right now, we're in that kind of 0.8 plus category. And you start to think about the performance of some of the genomic diagnostics that are out there on the market, where they're kind of 0.55, 0.6. We're offering a quantum leap here in terms of the level of performance that the technology can offer. And that's due to the nature of the biomarker, and it's also due to the nature of the platform.
So Irina asks a question.
Sure.
I think I have an idea of the answer. The number of autoantibodies is quite large. How do we deal with heterogeneity in the autoantibody array?
That's a very good question. How do we deal with heterogeneity using our current platform? I think that comes down to one of our applications. To be honest, Irina, one of the as I mentioned, we have a macro array, our IOM array, which has over 1,600 targets spotted in quadruplicate. So we have 4 readouts per antigen, and we've actually spent a lot of time developing that array with particularly heterogeneity in mind. And we've curated this array with specific considerations to biomarkers that are prevalent within cancers and autoimmune diseases, which have been taken from the literature. And so we've really tried to make it as cancer heavy as we possibly can in as many instances, and trying to address maybe some of that question. So there has been a real focus around that. And then I think as well, being able to drive applications which address heterogeneity. So for example, how can we look at intra-disease stratification? How can we start to really profile the spectrum and maybe some of the subpopulations within those diseases as well?
Thanks, Andy. And lastly, if anybody is interested in learning more or
Sure.
wants to get started, what's the process like?
Sure. So, I mean, I would firstly ask you to reach out to either myself or to Kevin. We also have an email address, which is support@sengenics, SENGENICS.com. If you have any questions, you can send us an email, and that will be automatically forwarded on to one of your commercial support guys within your geographical location, and they can then start to discuss and open up dialogue with you, whether it be some information you require, whether it be a question or a project or something to do with samples that you have in mind. We can certainly open that discussion. And then to perhaps give you a slightly more overt, direct answer, we would normally, set up that first meeting, and again, we can run through technology, but we can also really just start to understand what is your program? What are the things that you're looking to answer, whether it be specific to a drug or a disease, or something a little bit more nuanced. We really start to think about that and try and design an experiment which can support the question in mind.
Thanks, Andy. Are there any questions from folks? There was a follow-up from Xavier he asked if serum and plasma correlate then, which I think the answer is yes, based off what you said earlier.
Yes.
Any other questions? I haven't checked the Q&A. Can you detail, this is from Brad, can you detail how you design the arrays and the logic when constructing your arrays? He asked that a couple of minutes ago.
Yeah, sure. So we have a number of off-the-shelf arrays, as I mentioned, which have been constructed through the existing library we have with various considerations in mind. So we have a discovery array, which is spotted in quadruplicate, which has 1,631 targets on it. And that's a whole multitude, as I mentioned, of different autoantigens from different families and groups of proteins, which have been associated as being biomarker candidates within the respective disease fields. We then have some more tailored specific arrays, such as a P53 array, for doing the likes of functional restoration, where we developed that through an academic. So David Lane, P53 fame David Lane in Singapore, we have 109 clinically significant mutant variants with wild type. We have a cancer testis antigen array, which is really being used to address some of the questions, particularly the likes of IREs, which exhibit testis antigens prolifically. We launched that as a consequence of those types of requests, and also some other arrays as well. Like we have a pan autoimmune array. We have a COVID array. And we also have customized capability as well, to really customize array for your sort of liking. But the way that our content has been driven has been through organic growth, through request from collaborators. We don't just go out and produce content whimsically. There is obviously specific need which has been driven by the scientific community, and the way that we set our arrays up at this moment in time, I think we're able to run 4 samples per array. And again, we have high throughput screening capability as well in different types of formats. So we're not just bound to one particular design on our array. If we've screened, identified 25 markers we want to move forward through, then obviously we can put that into a much more high throughput screening template, which brings down cost, and it allows you to run much higher numbers and volumes of patient samples on the array as well. So I think we're always trying to bring down costs, and we're always trying to increase throughput as well.
So Savior wanted to know if you could use not artificial, so it's my own work, cerebrospinal fluid. Katie had answered the question, and yes, you can run samples of
Absolutely.
CSF.
Yes, definitely. Yeah.
So I want to wrap up. We're about a minute over. Thank you very much, Andy. Thank you, everybody on the call today
Thank you.
for joining. I'd also like to thank Camille, who set up the meeting today for us. He's in Kuala Lumpur and graciously has run the meeting and made sure everything was going smoothly for us. And to finish, I just wanted to quickly go through the poll question. When was the term antibody coined? It was Paul Ehrlich who did that in 1891, 1 year after the discovery that serum transfer from an infected animal could impart protection from that disease in an animal that the serum was transferred to. Now, these other dates are also pretty important. 1923 was the year that Michael Heidelberger determined that antibodies were polysaccharides, later proteins. 1940 was the year that the rheumatoid factor was discovered, which causes agglutination. So serum from rheumatoid arthritic patients can cause red blood cell agglutination. And 1948 was the year that Hargraves, using complement fixation, found the lupus erythrocyte cell. And those last two, the Waller test, Waller-Rose test, and the lupus erythrocyte, those were diagnostics to determine whether someone had those diseases going all the way up into the 1990s. New technologies replaced them, but that's where those numbers came from. With that, I'd like to thank everybody for joining us today, and I hope to see you at our next webinar. Thanks, everybody, and thanks again.